Okay, let's make a start. So, welcome to this session about the British Neuroscience Training Academy. So I'm Sandrine VM-Ravek. I'm the BNA Education and Engagement Secretary, and I'm also an academic at the University of Southampton in neuroscience and working on stem cells. So this British Neuroscience Training Academy has been launched a few months ago, earlier this year, and this is following some work we've done on the accreditation scheme that the BNA wanted to launch. Okay, so this is the program for today's session. So I'm going to tell a few words about the RSB Royal Society of Biology accreditation, to which the BNA contributed. Charlotte is going to introduce our BNA Training Academy and the different events that we've been organizing and that have happened already. Tara is going to give a taster session of a BNA masterclass on dementia. And this full masterclass will happen later this year in June, and you can register for that. We'll give you details later. Kate is going to do a session on science writing for a journal. She's the editor or co-editor of the BNA journal, Brain Neuroscience Advances. And Emma is going to give a little snapshot on taking first steps in neuroscience teaching. So these three talks from Tara, Kate, and Emma can give you a little taster of what to expect in the different training workshop events that will happen later this year and next year. So first, a little bit about the accreditation work that we've been doing. 2 years ago, I've been tasked to work on this accreditation scheme for neuroscience degrees in the UK. And rather than reinventing the wheels and create a BNA accreditation schemes, we decided to go with the Royal Society of Biology accreditation schemes, who had their program up and running, and add some criteria for neuroscience specific degrees. So we've gathered this working group and about 10, 12 people from really diverse backgrounds to think about what is it actually that we want neuroscience degree to cover, to make them a good neuroscience degree with width and breadth of coverage of neuroscience. And we came up with these 10 points, 10 learning outcomes, covering these 10 areas that we thought were important for a neuroscience degree to cover. And so out of these 10 learning outcomes areas, the first 4 are really specific to neuroscience, and they made it as learning outcomes for neuroscience degrees. The other 6 are a little bit less neuroscience specific, and we actually managed to incorporate them into the main criteria of the RSB criteria for accreditation. And we've now included some points on these 6 areas in the main accreditation handbook. So now the RSB is using this to accreditate all the degrees that apply for accreditation, whether they're neuroscience or not. And so this is now the 4 neuroscience-specific learning outcomes that have been incorporated into the Royal Society of Biology handbook for accreditation. And so you can see now they're using these learning outcomes when the degree in neuroscience is applying for accreditations. And so you can see they cover the core neuroscience topics. They cover some aspects of analysis and interpretation of experimental data, research finding and clinical studies. They cover some impact and translation of advancements in neuroscience, and the ability to describe emerging priorities in the field. So we thought these are really key aspects that students on a neuroscience degree should be able to explain, apply, describe. So that's all good, and it's been almost a year now that the RSB has incorporated these learning outcomes in their accreditation handbook, and we are going to, I guess, review what the experience of these has been, and talk to the RSB and get their feedback also on how this has worked this year. But we also welcome feedback from you. So whether you've had a degree going through accreditation or you're thinking about your degree going through accreditation, I think it's important we hear from you as well as neuroscience experts, whether this process is working, whether we need to tweak some of these learning outcomes, or whether we need to suggest to the RSB anything that they should do better or improve the general process. So you can just email me. This QR code is just linking to my email. Give some feedback. If you're a student through accredited degree, or if you would like your university to go through accreditation so you can have that stamp of quality approval, talk to us as well, because we can then work on that, put you in contact with your LGR in your local institution, and go from there. So this has been all well and good, but this made us realize that actually all these different learning outcomes, it's great for a neuroscience degree, but master students, PhD students, ECR, junior academics, or even senior academics also have training gaps that needs to be addressed. And so we identify different training gaps that could be filled by different ways, and that exists at different career stage in the neuroscience training of different populations. And also as BNA, as a national or even international institution, organization, we have the knowledge as a community of members to deliver some of that training, and we have the power to organize this and deliver these trainings. So this made us think about how can we leverage all this, and so we created the Training Academy. And so Charlotte is now going to tell us more about the Training Academy, what's happened so far, and what's to look forward next. Thank you.
Thank you, Sandrine. Hi, everyone. Good morning. I am Charlotte Shepherd. I'm Head of Partnerships at the British Neuroscience Association. Really pleased to be here today to tell you more about the BNA's Training Academy programme before I hand over to the rest of our panel, who'll be delivering some short taster sessions to give you a bit of an idea of the kind of training that you can expect from our new training programme. So just briefly, I'm sure many of you know that the BNA is a not-for-profit membership organisation representing the UK, but also the international neuroscience community. If you're not already a member, I would strongly encourage you to join. I'm not going to go into details about the many, many benefits of membership now, but please do go and talk to the team down in the exhibition hall to find out more about how to join if you're not already a member. A key pillar of what the BNA does as an organisation is working around our theme of inform. So that means to provide our members of all career stages and disciplines with the information and support that they need to advance their research and their careers. We do this in a number of ways, through events like the festival that you're all at the moment, through career development events, such as the workshops and sessions we were running in the career zone throughout the festival. also through offering bursaries, grants and prizes and running events across the year, and most recently through our newly launched training programme. So I'm really pleased today to be introducing the BNA Training Academy. As Sandrine said, we launched it earlier this year. Delighted to be joined by a fantastic panel who'll be offering kind of taster sessions of the kind of diverse training that you can expect in the coming months. So as Sandrine outlined, the BNA training programme has been developed alongside the BNA's Training and Accreditation Working Group and in consultation with our membership to address and identify the gaps in specific training in subject areas related to neuroscience. The goal is to develop and deliver a programme of training activity that will help build early career neuroscientist knowledge, expertise, and develop transferable skills that can support career development. All of our courses will be led by subject area experts, and they'll consist of online and in-person courses for neuroscientists across a range of disciplines, but also career stage and educational stage. And the training courses will range from one-day masterclasses, which will be in-person and online, right through to short one-hour webinars. Our training is open to everybody, but we also do offer discounted registration for our BNA members, and some of our training will be free to BNA members. So I just wanted today to give you a really brief overview of the kind of training that we've delivered to date, to give you a bit of a sense of the breadth of topic, but also the range of audiences that our training will cater to. In March, we ran a 2-day online course around image analysis. This was for primarily PhDs and postdocs, but also master's students, and it focused on the fundamental concepts and practical applications of image analysis, as well as giving an overview to participants of the applications of some cutting-edge neuroscience tracing tools. We've also run a short half-day introduction to neuroanatomy, so that was focused at undergraduate neuroscience who perhaps wanted a refresher in that area, but also first and second year medical students. And it covered the basics of brain and spine anatomy with some interactive discussion of clinical case studies. We've got lots coming up, so just looking ahead, in June, very excitingly, we have our first dementia masterclass. It's on the 4th of June in London. We're very fortunate to have our, as of yesterday, past president, Professor Tara Spire-Jones, and she'll be telling us in more detail about what that training will consist of in June. And the dementia masterclasses are sort of first in our series of planned masterclasses. These will be one-day training sessions where we'll take a kind of deep dive into different disease states, but take a really kind of multi-sector approach into how we look at that and explore the latest advances of research in that area. In June, we're holding a 1-hour webinar, which is free for BNA members, and that's looking at patient data and research from a data protection point of view, and looking at the principles that impact the kind of the use of patient data for research purposes. We're also planning an MND masterclass in September. That will take a very similar format to the dementia masterclass that you'll find out more about today. Location and speakers to be confirmed, so I would say do check out our website and our social media channels for more updates as we progress with plans on that. And then in November, we have a half-day online panel session, which is led by our colleagues at Lancaster University, and that's exploring the value of using different model systems in research and asking questions like, are we always using the best model systems for a scientific question? And to answer that question, are we missing opportunities to adapt our preferred models? Are there other approaches that we can use in other model systems? And can we break down silos to accelerate the impact of research in the way that we use model systems? So that will be a really great interactive panel sessions where participants can come with specific questions and challenges in their own research, and the panel can help to answer some of those. So that gives you, I hope, a bit of a flavour of the kind of the breadth of the training that we're offering. There's lots more in the pipeline, so I would say please do keep an eye on our social media channels, check out our website, because lots more courses will be confirmed in the coming months. We also would really like to get your feedback on thoughts. So if you scan the QR code there, it will take you to a short feedback form. We want you to tell us what's important to you, what's missing, where do you see the gaps, and what kind of training can we support, can the BNA support you with? Also come and chat to the team down in the hall at the BNA Donut. We'd love to hear your views, to answer any questions about the training. So I'm going to hand over to our panellists now to kind of take you through some taster sessions, but if you'd like to come and find me and chat to me about training, very welcome to. Thank you.
Yeah? Yeah. Okay. So if there are any pressing questions for Charlotte or myself on the new training program, we can take them now. Otherwise, there'll be opportunities for questions at the end as well after the three taster sessions, and of course, question on each taster sessions as well. Any pressing question yet?
Thank you. Is it working?
Yeah.
Oh, I can't hear. Anyway, I was wondering if you were planning any training related to bioinformatics or computational neuroscience?
Yes, so that's a subject that we already have in our list of interest, and we're going to organize that hopefully later this year, next year.
Yes, I think there'll be a lot more to come post-summer and into the next academic year, and that's definitely high on the list of topics.
I'm not an expert at all. I just would like to learn, which is why, I suppose, yeah. It's just something I'm interested in, so that's how I was wondering.
Yeah.
Thank you.
Yeah.
Thanks for this, was really helpful overview. Just a quick question. I wondered when you said that you identified training gaps, how did you go about that? What was the process for identifying those?
Yeah, so I think with our working group, we already we had an idea of in our day-to-day work where some gaps were. We also run a membership survey and ask for feedback. So there's a few areas identified there. And it's a continuous process. Yeah. So if you scan the QR code, and it will be back on the board at the end, do suggest to us any gap that you identify in your students, in your postdocs, in your labs, or even for yourself. If you're new to neuroscience and want more details on one area or the other, that would be of interest to us, so we can organize things.
Thank you.
Thanks. Any other questions? Okay, so I think we'll move on. So we'll welcome Tara for the dementia masterclass tasters. Thank you.
Great, thank you very much. I'm really excited about this training initiative, and we really do want to hear your feedback, so please let us know what you think. But as was mentioned by Sandrine and Charlotte, I will be running a dementia masterclass. These are my standard declarations of interest. Unfortunately, none of them make me rich. The registration is now open, so if you like this, we can show you the QR code again, but please do join us. This is going to be a full day, and it's going to be a very packed day, so what you're going to get now is just sort of a taster of who's speaking, what they're speaking about, and I'll go into a little bit more depth on my area of expertise as sort of a teaser. So the speakers are going to be Professor John Schott, Carol Brain, Henrik Zetterberg, Paresh Malhotra, Katie Lunanen, and Dr. Laura Adram, our very own CEO. And if you're not in the dementia field, trust me when I say this is a star-studded lineup. You can ignore me, but the rest of them are really, really impressive. So it is real experts in the field telling you about their area of expertise. And who we're aiming it at is sort of anyone who is interested, but we think it would be a really nice introduction to people who might want to come into the dementia field. If you know of clinicians who treat dementia patients and just are interested in the research, they might be interested in this. And so we hope it'll have broad appeal. So this is the program that we have so far. I won't read through it to you, but we're going to do these lectures from experts in the field, but putting in time for discussions. And at the very end, we'd like to have a workshop where we want to have some interaction about what people think are important gaps in dementia research, what we should be thinking about moving forward. So we want the participants to also help us as dementia researchers in the field and discuss what problems you think are important. So I'm going to start out by telling you a little bit about, this is the teaser part, so about the dementia research space. And you might be surprised to learn that the idea of dementia has been around for 4,000 years at least, I suppose since people have lived long enough to have age-related cognitive decline. And when I was looking into this, The earliest record I could find mentioning something like dementia was actually in a papyrus scroll in ancient Egypt, so 4,000 years ago. I can't remember the name of the scroll. Of course, they didn't call it dementia, and they didn't know it had anything to do with the brain, but they described a perishing of the mind and forgetfulness, which I think is still a beautiful way of summarizing what happens to people as we age. And then in about 1000 BCE, or before Common Era, the ancient Indian texts mention not only symptoms of cognitive decline and aging, but also some herbal treatments to try and help people with cognitive decline and aging. Plato described cognitive decline symptoms similar to what we would call dementia today. The ancient Chinese described this in their early medical texts. Hippocrates and Galen, so other famous ancient Greeks, actually were the first ones that I think I could find that thought it might have something to do with your head. So they attributed dementia symptoms to the brain. And then there was this long period in the Middle Ages of sin and mysticism, and as far as I can tell, not many people were thinking about, or at least not writing about, the idea of cognitive decline or dementia and aging. So there was I guess they had bigger fish to fry. They had the Black Death was killing them off probably earlier than you could get dementia. So this is the dance of death. There was this beautiful period in art where people were obsessed with death. But modern dementia research really kicked off in the early 1800s with French psychiatrists Pinel and Esquirol, and they made the modern definitions of dementia. So I'd imagine all of you have you all met someone living with dementia? Is there anyone who has not ever met someone? No, it's super common. So you all probably are familiar with the basic idea. Dementia symptoms are this progressive loss of cognitive function from your normal baseline. And that's what these two early neuropsychiatrists defined. And so that's when we started actually trying to think about what's happening so that we could eventually treat it. And in the late 1800s, there was an explosion in technology. Things like microscopy, formaldehyde fixation, ways of staining brain tissue so you could actually look at it. And that allowed people like Oscar Fischer, Alois Alzheimer, and Solomon Carter Fuller to look at the brains of people who had the symptoms that we're all too familiar with. So interestingly, most lectures on dementia will start with Alzheimer. So if you get a little bit of history, it starts with Alois Alzheimer and August Dieter. But I think it's really important that we realize that there's a broad multicultural history of dementia research. And even in that period when Alzheimer described the first patient of the disease that later became named after him by his mentor, there was a lot more going on. So Oscar Fischer actually was the first person to describe one of the key pathologies, which are called plaques in the brain, and I'll show you some in a minute. So they were called Fischer plaques for a long time. Alois Alzheimer and his student Solomon Carter Fuller described both plaques and neurofibrillary tangles that I'll show you in a moment in the brain, co-occurring in people with dementia. And it's just a fascinating history of why this disease ended up being called Alzheimer's disease and not Fischer's disease. Part of it might be because Fischer was killed by the Nazis in a prison, so he didn't get a chance to have a long career. But another important figure that we don't talk about too much is Solomon Carter Fuller, who was the first black neuropsychiatrist in the United States. He trained with Alzheimer, and Alzheimer described a single patient, August Dieter, initially, and that ended up in a textbook by his boss. But Carter Fuller actually made this into a bigger field and looked at lots of people with dementia and was the first person to describe these pathologies in people with normal aging, not just with disease. So fascinating history that we can go into if you're interested. And then it wasn't until the 1980s that we started to get a handle on the molecular mechanisms underlying these brain pathologies that were described in the early 1900s. And then it wasn't until the last few years, 2023 and 2024, when the first disease-modifying drugs were approved. We've had a few symptomatic treatments since the 1990s, but only in the last couple of years have there been drugs that can slow the actual progression of the disease. So it's taken us 4,000 years to get there, but we are finally making progress with slowing and hopefully eventually actually meaningfully stopping or preventing these diseases that cause dementia. But dementia is the symptoms that we're all familiar with. What Professor John Schott is going to tell us about is what clinically these are. So he's going to talk about the different causes of dementia. He will tell you hopefully about the prevalence of dementia, which, as you know, since you've all met someone, is really growing as our population ages. And John is also the Chief Medical Officer of Alzheimer's Research UK, so he'll be able to tell you a lot about the lived experience as well. We're going to have a lived experience partner. I think it's something we really do need to think about, even as people like me, really fundamental neuroscientists who cut brains up in a lab. It really is inspiring at the very least, and I think important in framing our research questions to know what the experience is of people living with these diseases. So we're going to have lived experience partners. Alzheimer's Research UK is going to help facilitate this. Then we'll have Professor Carol Brain, who's one of the real powerhouses in understanding how lifestyle factors and sort of things that we're exposed to can influence our brain's aging. She'll talk about the causes of dementias in terms of risk factors. I think about these as age, the most important risk factor, genes, which we heard if you came to Julie Williams' amazing lecture yesterday, you have a really good handle already on the genetics of Alzheimer's disease, and lifestyle factors. And Carol will be able to tell us some of the amazing data about how not only do we have these drugs now that are far from perfect, but because of an understanding of the risk factors and things like introducing the smoking ban and pushing education, we actually are seeing not a reduced prevalence of dementia, but a lowering of the individual risk because we're making our brains more resilient. So there's some good news in that space as well. She'll talk about these modifiable risk factors, I'm sure. So this is the Lancet Commission's paper, and I always talk about this because it's this massive meta-analysis that Jill Livingston and colleagues put together every few years for the Lancet Commissions. So it's really solid evidence, epidemiological evidence of modifiable risk factors for dementias. It's important to understand that you can't know that these are causative just because there's an association, but the data are coming along, and it gives neuroscientists like us a really good idea of what to go and look at in the lab, what to try and understand. So the 14 factors that are really well substantiated to be associated at least with dementia risk are less education increases your risk substantially, untreated midlife hearing loss. This one was a surprise to me when it first came out a few years ago. We don't know why this is the case, but if you get hearing aids, this seems to take away that increase in risk. And my best guess is it's about keeping your brain active and resilient and engaged, but we don't know that for sure. There's a lot of research in that space. Smoking is bad, and you might have heard the Parkinson's lecture where smoking is associated with reduced risk of Parkinson's, but for just about everything else, it's bad. And that's the case for dementia. High LDL cholesterol is associated with risk, and there have been some really interesting and high-profile papers and news stories coming out recently about treating this and how that is sort of lowering your high blood pressure or your high cholesterol is really getting more and more evidence that this is maybe a causative factor and not just an association with your vascular system. Depression is associated with increased risk, traumatic brain injury, physical inactivity. There's a whole list, and I'm sure Carol will give us lots of the latest science on how all of these things are thought to be affecting our brain's resilience as we age. But the take-home message for me from all of this amazing data is that over a third, maybe up to 45%, almost half perhaps, of dementia cases could be prevented. That's all-cause dementias. And I really I think it was your question, Rick, about how did the math adds up to Julie Williams if 45% of dementias could be prevented according to this meta-analysis, but 90% is genetic according to the genetic analyses. Where is that? Part of it is these data are all-cause dementia, not just Alzheimer's, right? So I think the numbers will be a bit different there. I think there might be a heavier genetic component for pure Alzheimer's. Some of these are a little bit heavier on the vascular dementia side. But the real answer is we don't know yet. These are all our best guesses from different angles in the field, and they haven't really, as Julie said, been put together properly yet. So we'll have the real experts to try and fight that out a little bit at the masterclass. Henrik Zuckerberg will talk about biomarkers for dementia. This is a really hot topic in the field because we know now that the pathologies are building up in the brain for at least 20 years before you have symptoms. And most of the treatments that are being developed, the pharmacological treatments, are much, much, much more effective if you can get them in early before you already have neurodegeneration. But we need a way of knowing, because dementia is this overlapping set of symptoms between lots of different underlying causes. So if somebody is in the early stages of a sort of dementia with Lewy bodies, you don't want to be treating them with something that's designed to treat Alzheimer's, because that would be ineffective and has potential side effects and harms. So the biomarker space is really coming along and getting the ability to identify people who have Alzheimer's-like pathology very early on in the brain. We're best at Alzheimer's so far, less good at the others, although you might have seen the alpha-synuclein you can now detect with something called RT-QuIC in your CSF at least. So we're coming along with the other causes of neurodegeneration and dementia. But Henrik will tell us hopefully a lot about how this space is going. There are even blood assays. You don't have to take bits of brain, which is what we've always had to do in the past to confirm your type of dementia. You had to either wait till you die or donate a bit of brain, which nobody's keen on, to actually go and look. But now with blood and CSF and imaging biomarkers, we're really able to start narrowing down, and that's super important for the field. And then I'll be talking about advances in the neuroscience, because that's my space, this is my comfort zone. So I'll talk about how these risk factors, age, genes, and lifestyle factors, are contributing to the brain changes that we see, the protein aggregation, the loss of synapses in neurons, and the sort of downstream mechanisms that are leading to neuron death, and how, of course, those are the things that actually cause the symptoms that we see. So I'll talk about Alzheimer's disease, probably for most of the time, because we don't have time to do everything in the masterclass. So I'll probably focus in on Alzheimer's disease, talk about the cardinal features. So these are the Fischer plaques. So you remember Oscar Fischer? So he described amyloid plaques. We learned in the 1980s that those plaques are made of a sticky protein called amyloid beta that clumps outside the brain cells in the brain parenchyma. The other cardinal pathology that was described by Alzheimer and Fuller was the tangles, and the tangles are inside the neurons. They could see them with silver staining, but in the 1980s and 90s, it was 90s for tau, I think, we finally found out that it's hyperphosphorylated tau protein that clumps inside the brain cells, inside the neurons themselves. And those are much more associated with neurodegeneration. And the third feature, so it's plaques, tangles, and atrophy, which I'm showing you at the top. So you can see that the brain is just shrinking. Alzheimer's Research UK has a lovely metaphor. It's that you lose about the mass of an orange from your brain. That's why their color is orange by the end stages of Alzheimer's disease. And those pathologies, as I mentioned, don't show up all at once, but they accumulate very, very slowly over the period of the disease. So the prodromal phase in the middle there is when you are perfectly normal, no matter what cognitive test you take. You don't have any symptoms whatsoever, but even in that prodromal phase, if you're on the path to Alzheimer's disease, you're already accumulating huge amounts of amyloid beta. You'll already have some tau pathology in your brainstem and medial temporal lobe. And at about the time when you have detectable symptoms, mild cognitive impairments, you start to really ramp up the tau pathology, start to really lose the neurons, and those are the things that really are driving the cognitive decline, is the loss of the neurons and loss of the synapses in between. So what is amyloid? Amyloid plaques are made of that sticky amyloid beta monomer. It's generated from amyloid precursor protein, and you might remember from Julie's talk that that's one of the genes that's mutated that can cause familial Alzheimer's disease. And the other two that can cause familial Alzheimer's disease are the scissors that chop the amyloid beta peptide at one end. So there's strong evidence that amyloid is an initiating factor in the disease, at the very least in familial forms, but most of us in the field are pretty convinced that amyloid is an early part of the disease process in the more sporadic cases as well. So those amyloid beta monomers are, they're part of your brain. They're there even if you don't have Alzheimer's disease. We're not entirely sure exactly what they're there for, but they have some functions in synaptic plasticity. Some, Rob Moore described them as an antimicrobial peptide, but in disease they stick together into these oligomers and then fibrils, and then the big plaques that Fischer and Alzheimer and Fuller could see in the brain. Tau is a much more complicated protein, and my favorite protein, actually. So tau is encoded by the MAPT gene on chromosome 17. I've got a cartoon of it here. It's not for you to be able to read from the back, but all of these tiny little lines coming off of it are mutations or variants in the tau gene that are associated with disease. They're not associated with Alzheimer's disease. They're associated with frontotemporal dementias and other types of primary tauopathy. But tau does aggregate in Alzheimer's disease as well as in the primary tauopathies. And when the tau gene is made into the tau protein, we have 6 isoforms in the brain that are different by these pink colored exons here. So the differential splicing of these exons causes these 6 different isoforms. And importantly, exon 10 inclusion makes tau have 4 microtubule binding repeats. And that gives you a hint to its normal function. Tau is normally a microtubule binding protein, sits along the microtubules in the axon and helps keep them stable, as well as having many other functions. And changing the isoform changes its propensity to bind the microtubules, and it also changes its propensity to self-aggregate. And tau, much like amyloid beta, it's of course made in 1 copy at a time. It becomes phosphorylated as part of its normal regulation. In disease, it becomes aberrantly phosphorylated, sticks together in oligomers, makes these filaments, paired helical filaments and straight filaments, and ultimately clumps up in the neurons as tangles. And what's really come out of the field since I joined, which was many years ago now, is that it's not really those big lesions that Alzheimer and Fuller and Fischer described that are toxic to the brain, but these soluble forms of the protein, somewhere between a single copy and a huge lesion, is what's more bioactive and what's actually causing the damage in the brain. Oh, it's not moving. There it goes. And this is just self-indulgent, frankly. I was on an electron microscope a couple of months ago, and for the first time in my life, I was looking at paired helical filaments, which have been described since the 1960s. So this is a tau tangle inside a human brain electron micrograph, and they're just so beautiful, the paired helical filaments. So this is just an excuse, really, to look at them on a big screen. So the other thing that I'll talk about a lot in my part of the masterclass is synapse degeneration. That's what I work on, so I have a bit of a bias there. But it is an established fact, long before my time, that synapse loss is the strongest pathological correlate of cognitive decline in Alzheimer's disease. So better than plaques, tangles, inflammation, atrophy, anything we can measure, the loss of synapses is the thing that predicts your symptom progression the most. And what I'm showing you again is just because I love EM so much. This is now 3D scanning block face microscopy, so it's a tiny, tiny bit of human brain, 30 microns by 30 microns by 30 microns, and we're going through 50 nanometers at a time, and you can just see how amazing your brain is, how busy it is. There's not a single nucleus in this image. All you're getting here, you can see the myelinated axons, you've got synapses, dendrites, spines, myelinated axons, and that's just a tiny, tiny portion. So you have 100 trillion synapses in your brain. And so that's one of the reasons that it's been so hard to crack Alzheimer's disease, is because the brain is so complicated. But it's also a really important part of the field that we're looking into how synapses die, how they might be involved in disease pathogenesis. We'll also talk about non-neuronal cells. So I trained as a synapse biologist, and I love them very dearly. But as Julie Williams pointed out really elegantly in her talk, there is a lot of evidence from the genetics that the disease process in Alzheimer's is driven by microglia or other glial cells. So there's a huge part of the field looking at how all of these different cell types are contributing to neurodegeneration in Alzheimer's disease, and I'll spend a little bit of time talking about that. This is a review that we wrote a couple of, 6 years ago now. So if you want to read a little bit more about the glial roles, this will now be quite outdated. So pick a more recent review. But we've got a couple of others, but I still like the cartoon. And the other thing that I'm really excited about as a scientist, so I'll probably spend quite a bit of time on it, is the idea of how these pathological proteins move through the brain. So I showed you that they accumulate very slowly over time, and in terms of where in the brain they are, they also start in certain regions and then spread out. And so one of the things I think is if we could stop that spread, especially of tau in Alzheimer's disease, we could probably stop disease progression, because wherever tau goes in the brain, neurons die, and the spread of tau pathology through the brain correlates almost as closely with cognitive decline as synapse loss. So it's another very strong predictor of cognitive symptoms. So what I'm showing you here is Alzheimer's disease and a primary tauopathy called progressive supranuclear palsy. So even in non-amyloid containing diseases, tau starts in certain brain regions and moves through the brain. And one of the things we're doing is trying to figure out how that's happening and how we might stop it. We have evidence from our lab and from many other labs around the world. This is one of those situations where three independent groups published the same result at the same time in 2012 in mice, that pathological tau can spread through the brain by jumping from one neuron to another through their synaptic connections, so jumping from one brain region to another. So this is a beautiful image from PhD student Caitlin Davies when she was in Edinburgh with me, where she injected human tau in the entorhinal cortex, which is on the far side there, and the human tau is labeled in red, and the virus-expressing cells are labeled in green. And we could use technology like this and like transgenic restricted lines to show, in mice at least, that human tau jumps through the brain by leaving the entorhinal cortex and entering the dentate gyrus molecular layers, the connected cells. But the question that we were interested in recently was this happening in humans? Because in these mouse models, we are overexpressing human tau a lot, like 13 times normal. So we could be driving it out of the cells just because of the system. So we developed a technique that I based on Steve Smith and Christina McCabe's array tomography technique to be able to look inside individual human synapses. So we were able to look at whether synapses contain tau in human postmortem tissue and start to get hints of whether it might be spreading. But keep in mind, human postmortem tissue is a snapshot at the end stage of the disease, so we can't prove that it's actually moving with the postmortem tissue. So I'll also talk a little bit about how the new disease-modifying treatments for Alzheimer's disease work. These are so far all targeting amyloid beta, either the fibrils or the plaques themselves, the bigger fibrils. And these are antibodies that you inject intravenously or infuse intravenously. And unfortunately, although they do slow disease progression, and we had a lovely talk by Jeff Kirchner about this, right? They do slow disease progression by about 35-40% in the right populations, but they are very expensive. And by very, I mean maybe 30 grand per year per person. And they come with high risks. So some people who took these drugs on the clinical trials have died because of the brain swelling and edema, the ARIA-related symptoms that Jeff mentioned. And you have to have these MRIs every so often when you're treated to watch out for these symptoms, so it's incredibly expensive to deliver. So even though the UK's healthcare regulator, the MHRA, approved this drug and said it's safe and it is effective, it's passed its phase 3 clinical trials, here in the UK we can't get it on the NHS because the risk balance reward is too poor. Right, and I completely understand. I'm glad it wasn't my decision, but because people were very disappointed to not be able to access a potential treatment for dementia, but only slowing the disease moderately and coming with this many risks, I can understand completely why this decision was made. So then at the end of the session, I'll, of course, provide plenty of papers if you want to go away and ask questions or do further reading. And I'm really hoping that we'll have a good discussion at the end about what people think about how we should move forward as a field. So that's just the teaser. Thank you. And I'm happy to take any questions on the research or the training session if you want now or at the end. So to you. Also happy if there aren't questions. Maria? Yes, Charlotte, how big is the venue? 100? Yeah, I don't think we're full yet, though, right? So, okay, so there should be places left, and like I say, we're hoping to make this an annual dementia masterclass if there's interest. And one of the things we'll do at the end is ask people of this. We'll show you this broad range of what's going on in the field, and ask: is there a particular thing you would like to have an in-depth day on? Do you want to learn everything there is about biomarkers or everything there is about imaging or whatever aspects, risk factors? So think about that if you do come, or if you're coming to any of the master classes, you can help us tailor them. I don't know if we're going to film them, but that would be another thing we could, if we had the technology and could afford it, we could put it online, but I'm not sure if we, yeah. Anything else? Great, well, I'll pass over then, thank you.
That's great, thank you, Tara. So I'm delighted to welcome Kate Baker as our second taster session. So she's going to talk about scientific writing for a journal. And as far as I know, this is not scheduled yet as a full training sessions. But if you're interested in that, let us know in the feedback, and we can plan that for the autumn or the winter, or at some point next year.
Absolutely. Thanks, Sandrine. And wow, that was fantastic. I'm just imagining if 5 or 10 of those 100 people, for them, if that day is a pivotal moment that is a part of their roadmap in career decision making, it will be really impactful. So what I've had the opportunity to do today is put together a skeleton of the kind of generic cross-cutting training activity that can be included in the training program. And really, really keen to see if everybody from different perspectives thinks this would be useful, and then we can think how to expand it into a full offering within the training program. And really thinking about how what the BNA and the Training Academy can do can be complementary to people's local training and experiences, but really augment that and sort of network the training community into the BNA and support their impactful science and their career development. So this is just an idea really today of how we might do that and focus it on science writing for a journal. My name's Kate. I'm a geneticist and a cognitive neuroscientist based in Cambridge, and I have the pleasure of being the editor-in-chief, one of the 2 co-editors-in-chief of your journal, BNA Advances. So a little bit to ask you a question, first of all. This is intentionally blank, just to get a feel for who I'm training today. How many of you have been part of a co-author team on a published article? So in any respect? Some people, but not everybody. That's great. How many people have been the lead author on a paper? And by that, I mean not necessarily the first or the last author, but the person who's coordinated the process, pulled all the information together, and done the really substantial amount of the writing of a paper. How many of you have led a publication? Yeah, and so slightly fewer, and that's what I was hoping to do. And broadly, show of hands, if you're thinking, any of you, whether you've done it before or many times or never before, if you think about writing a paper, either as a co-author or as a lead author. Are your feelings one of kind of optimism and excitement and a part of your work that you really look forward to and enjoy? I'll put my hand up for that. And how many of you, and obviously there's a spectrum, there's a dimension, how many of you, if you think I'm now going to write a paper and publish it, feel a slight sense of pessimism or even dread or anxiety or anticipation, or maybe a mixture of the two? Yeah, that's normal. I think that spectrum is normal. And I hope to shift some of you a little bit on that spectrum today toward looking forward to it, enjoying it as a critical element of scientific practice and a creative opportunity that can be fun and really rewarding. So great, that's the blank slide. So when you're involved in a piece of work and you think, right, now I'm going to write the paper, it might feel like this blank sheet. Where do I even begin? Is this a new start, a new process? And what I put together is a little bit of a map, an idea about how it's not a blank sheet. And the map is, hopefully, snakes and ladders board. Okay, so it's not a game, but let's use the snakes and ladders board as an idea. Okay, so I'm going to take you through some steps in this process along the board to give you a little bit of sense so that it's not a blank sheet. So when you're playing this snakes and ladders game of writing and publishing paper, where do you begin? Well, step 1, and it's also step 100, is the bigger question of finding something you really want to write about. That is, see if this is going to work, both step 100 and step 1. It could be a whole other training session or a whole other discussion. So whether it's a review article or an opinion piece or a piece of original research, if you don't care about it, if you don't think it's important, if you're not interested, it's going to be really, really hard work. So that, I think, is always step 1 and step 100 in any piece of written work. Step 2 is to plan and do the science. Again, I'm not going to talk you through that today, but the key thing in terms of writing is that the article, the part of the finished product of a study, emerges as you're planning and doing the science, and you develop the writing as you go. And how might you do that? Well, whether you're a student and an undergraduate writing a project proposal, or a graduate student putting together a PhD proposal, or a PI writing a grant, that's actually all part of the process of developing the writing as you go. When you write your ethics applications, then there are more formal ways that you can write parts of your paper, the end product of a study, as you're doing the science. And that might be by pre-registering. So the process of pre-registering within Open Science is that you may write the background, the introduction, the methods, the planned statistical analysis, your predictions, and publish that within the Open Science framework or on other platforms. So you've actually written half the paper before you do any of the science. That's not essential for all studies, but it can be a way that you develop the writing in the article as you go. And an even more of a formal way of writing a study as you're conducting the science is to do a registered report. And that's something that perhaps within an Open Science session on the training program you would come to understand. Again, it's not critical for every part of neuroscience, but in a registered report, you would submit essentially that pre-registration to a journal, or have it reviewed, have your study design reviewed, have it accepted in principle by a journal before conducting the science. So you've written half of the article and had it accepted as you go, and that's one potential writing process. But even if you don't go through those formal processes, every time you put together a presentation, you're structuring the research questions, the background, you're developing the figures for your paper, you are writing your paper as you go through other aspects of the activities within a project. And something like a poster, some of you may have posters in the conference today, and putting together a poster, getting feedback from it, from your team, from conference attendees, is a really important step of developing the writing of an article. So, how does that look on the snakes and ladders board? You've planned and done the science, you've moved up some ladders, particularly if you've done pre-registration and registered report, you've written a good bulk of the article. And you're approximately halfway up the board already, rather than getting to the end of a project and then writing the article. So that's 2 to 21 to 42. Step 43, well, you've now completed the science. Whether it's a review article, whether it's original research. What are you going to do next? So there's 2 steps next, little steps. One is, well, choose the article type that matches the work that you've done. And I've put up here the current article types offered by your journal, Brain and Neuroscience Advances, just as an example. You know, have a really think through, what are you trying to communicate? What's the right kind of format, the right length? Within original research, we have short reports and we have longer articles. All journals will have a range of article types, and have a really good discussion within your team or with your supervisor about what's the best match to the science that you want to communicate. That's a little step. So you go one step forward on the board there. And then the next step, and these go hand in hand. Some people ask me, which do you do first? Do you choose the journal? Do you choose the article type? They go hand in hand. I personally prefer to think, well, what do I want to write? What's the right structure and length of the article? And then find the appropriate journal, but some people do it the other way around. So choosing target journals could be a long discussion, and I'm not going to sit on it for too long today. But the way I approach that is to think, what's the context, the scientific context, which best matches the science that I want to communicate? What are the other papers where, if you were to look at a journal contents list, where my paper would be best cited around its sort of nearest neighbours? And that has something to do with who do you want to communicate your science to? That could be a whole longer discussion. That takes you there. So you're sort of in the middle of the board, right? You've made a couple of decisions. And the important thing with target is journals. You know, most of the times that I've published something, it has been sometimes pitched to one journal, and an editor will consider whether they think that it's a good fit for their journal, and it'll come back to you and you move on to another journal even before it gets peer reviewed. And that's a totally normal part of the publishing process. So I think when you do this for the first time and you submit a paper and it gets returned to you or rejected by an editorial board, it can feel like a big setback. But I think just expecting that that's a very normal part of this process of matching what you want to communicate with the priorities of a journal, and not to be surprised or demoralized by that is important. Okay, and you might choose Brain and Neuroscience Advances. There are lots of reasons why you would choose our journal, your society journal, but it's just one option. We're really keen to, I'm keen to talk to you afterwards if you've got work you might want to submit. Then you're just going to draft the paper, next step. Now, obviously, if this was a longer training session, we would talk about this in a lot more detail. You know, how do you do that? You've got files and files on your computer with presentations and previous writing and results and methods and figures. Where do you begin? And there's no one answer to that. People do it in different ways, and different articles are written in different ways. Sometimes, for an empirical study, you might begin with organising and structuring your methods and presenting your results. You might even start with organising your figures and your tables. Sometimes people leave that to be the very last thing they do in the week before submitting the paper, and that's fine. But actually, that core material might be what you want to spend time on developing and structuring and getting feedback on at the very beginning, and then to build the writing of the background context of the introduction and then reflecting on your results in the discussion. You might want to do that at the end. There are articles I've written like that. And there are other ones where you start at line 1 and you want to write it from the beginning onwards, and that might be because it's a perspective and opinion piece, or because that's the way you're thinking, that you really want to set it up. I would say, irrespective of that, the advice of what I do with my group members is the critical nugget of any paper is a clear definition of either your objectives or your research questions. You know, the way where they sit, whether they start at the beginning of the paper or the end of your introduction. For me, I think, you know, I spend a lot of time being able to articulate what the objective and the questions to be addressed are in that paper, even if it's a review article or anything else. And I keep coming back to that as I'm writing the paper. Am I really answering those questions, and am I structuring it to address those questions? So I guess if there's one bit of advice, I would be spend a lot of time on nailing what those research questions are. Okay, but you draft that. So on my Snakes and Ladders board, that's a reasonably sort of short middle part of this overall Snakes and Ladders. But it can take a very long time to get to the stage where you've got a full draft of a paper. There are things I've written in a week, and it's been a sort of a joy and a creative process to start at the beginning and write something from beginning to end. But that's been quite unusual. And don't worry if it takes a long time, because it can do. So you've done that, you've drafted a full draft of the paper. What are you going to do next? Well, you might be a sole author, but mostly you'll be working with a collaborator or a supervisor or your whole team of scientists. They may be within your group, they may be spread across institutions, and now begins a process of feedback and revise. And my group will certainly say, they think they've drafted a paper, they're poised to submit it, and they're astonished at how many times we might go round the snakes and ladders of a circle there, hopefully with ever-reducing amounts of change with each cycle of feedback. But not always, because sometimes in the process of looking at the logic of your studies and your experiments and presenting the science, you might collectively realise that there is a missing aspect of logic, or an aspect of analyses that you're not confident about, and you might need to go back and consider doing a new experiment or including more participants until you're really confident in the science that you're going to put out there on the record. And that's absolutely fine. I've done papers where I thought I was ready to submit, and actually after a round of review realised there was another year's worth of work, and being brave about that something I'm quite proud of, and I ended up being able to publish an article that with a particular co-author that we're exceptionally proud of, by being brave and realising we needed to increase the response to feedback and go back again. So again, anticipate that process of many rounds of improvement until the work is collectively as strong as you can make it, but not perfect, because nothing's ever perfect. So the next little step is to prep and submit an article where you feel, right, now we're ready to send this to an editor, get feedback from editor and a feedback from peer review. There's definitely a session we could do on the peer review process from a peer reviewer's and an author's point of view. And do take time where you've put years of work and thought into a study and into all this writing, and you're so desperate to upload it and submit it. But at that point, you really are placing a potential product, an output to a publishing platform, and do it justice by producing something that a peer reviewer is going to enjoy reading in terms of its formatting, in terms of typos. Again, nothing's ever perfect, but don't rush that upload stage. Give yourself the best chance by submitting some really high quality product. Step 56, we won't, and then you wait. Now, how long do you wait? My record for shortest time of waiting for a desk reject for an editor was about 2 hours. That was, I submitted something ambitiously to BNA, and it's amazing how quickly they rejected that. So that wasn't a long wait. But you may find that you wait weeks or months for an even initial decision about whether a journal is going to send it out for peer review. I quite enjoy that period of time of waiting, because in that period of time it's out of your control. You can think only positive things about the outcome you might get. So you wait, and maybe then you receive and respond to reviews, and we can have a whole session or part of a session on that. Now, the reviews may think, they recognize the contribution you're making, they make a few sensible suggestions about making your article clearer or improving the figures, ask you to expand part of your discussion or limitations, and you, within a week or two, you can make those minor revisions, and suddenly you're up the ladder whereby your paper is going to be re-reviewed by an editor, accepted and published. It may go like that. Or it may be a bit different. You may, wait, where have we gone? Where's my little person? You may wait, and your hopes get higher and higher, and you're anticipating this celebratory experience, but in actual fact, some reviewers have really thought through your paper. They've put a lot of time into thinking about the experiments and the results, and they've said, we recognize that this is going to be worthwhile science, but as it happened to me a few times, they thought, well, there are some really key elements of evidence that's missing, and we think this isn't ready for publication yet, or perhaps it's not robust enough in how you've demonstrated, and you go all the way down the snake back into do some more science. And that, again, can feel like a really big setback. and it's something that you need team support and planning and how to respond to. But keep going, because actually you will get there, and it takes years sometimes. But again, my experience has been that that can result in work that is more impactful and ultimately a creative product and output that you feel even more proud of as a consequence. So, whichever of those sorts of routes around the board a paper can take, finally, you will get that acceptance. There is some justice in this system. It's not just random. There is a process by which a really well-formed output of scientific writing will be accepted for publication with that credibility and that stamp of peer review and all sorts of other things. And then there's just like last steps to go through about things to do with paper licensing, open access, article processing charges, which you need to think about before you submit, checking proofs, which I find quite stressful. So you have one opportunity to try and spot the copy editing mistakes that creep in, and you think, how is there a system that manages to introduce errors that weren't there in the first place? And step 100, that thing you cared about, you thought was important, that you spent so much time working on as a team, makes its way into the published literature. Gold medal for you. Okay, so those are the steps. Number 1, find something you want to write about. 2, plan and do the science, develop the writing as you go. Choose the right article type. Choose a target journal or journals, which are the right platform for your work. The process of actually drafting the paper, leading that, coordinating it as a team. The loops of feedback and revision, which can be grueling, but are actually a collective creative improvement process. Prep and submit a high-quality product that's going to be a pleasure and easy to peer review. Wait. Receive and respond to reviews. And then your paper will be accepted, licensed, and published. I don't like snakes and ladders as a game. There's no strategy in it. It's just roll the dice and move and see what happens. It's not the kind of game I find satisfying. And that's why actually I don't think that Snakes and Ladders is the best game analogy, although it might be useful to think about and understand the process, because it does involve some luck publishing your work, but there is, as I say, some justice and some logic in it too, in my experience. It can be frustrating, but overall, it's a process that I think is good for science and good for science careers. Luck and justice come into it. That's the little framework that I put together for today. I don't know how that was time-wise, but I'm really happy to take questions or reflections now, talk to you afterwards about work that you'd like to publish, and I'll be on the Sage stall with Nisha afterwards to talk about the journal. But also, maybe afterwards, you can think about whether this sort of content or skills development workshop with some interaction and sharing experiences could be the type of session that the training program might like to develop. Hope that was okay. Thanks, everybody.
Thank you, Kate.
Okay.
Any questions for Kate?
Okay, a quick question for me, from a training thinking point of view. How many either students get some sort of training on how to write papers or the publishing process within your degree programs or graduate programs? That would help us to know whether it's needed. So if you've had, nobody's saying they have had it. Okay, that's helpful. Maybe at master's level. Cool. Okay, that's helpful to know.
Yeah, I think that's really useful. I mean, we could develop at least an hour session on each of these 10 steps.
Really a workshop interaction.
Yeah, that's great. Thank you. So it's my pleasure to introduce our third speaker on a taster session, and that's Emma Wynell from Cardiff University. Emma is an expert at science communication and neuroscience teaching with different awards, so we're very lucky to have her here. And again, this is at the moment not planned for a full session, but I'm sure Emma would be happy to contribute a full session, half a day or even a full day online or in person on some aspect that she's going to tell us about. Thank you.
Thank you, Sandrine. Morning, everybody. Hope you're all well. As you've heard, I'm Emma. This session came about, Charlotte and I were having a chat about teaching in neuroscience. I'll go through a little bit of a career history to help you understand kind of the angle that I come at in discussing this. But actually, I think there is potentially a gap in developing something around neuroscience teaching, whether that's taking your first steps into neuroscience teaching, or maybe people who are a bit more established in their teaching as well. So this is a bit of a timeline for you, and I actually always feel a little bit uncomfortable describing myself as a neuroscientist because I started off in these glasses, apparently, doing a biochemistry degree. This is in my undergraduate time, because I'd actually never really come across neuroscience before at school. I really loved biology and chemistry, so I just kind of mashed them together in a biochemistry degree. It was only a little bit later that I was given the opportunity to do a PhD in behavioural neuroscience. So that's where the neuroscience link kind of comes in. And I kind of worked my way up the career ladder, I guess, going through a fellowship, which was more clinical, lots of neuroscience research. But it was kind of during this time that I came to the conclusion that although I loved research and really value it and see the importance of it, for me, the educational piece and the engagement piece and the outreach work aligned much more clearly with my values, and I was probably better at it, to be honest. So I became a lecturer in 2019 on a teaching and scholarship focused pathway, so you may or may not have that if you're working at a university. So what that means practically is I do lots more teaching, lots of research into education, and there is a poster actually on some educational research we've done in the next poster session, just a little plug for that there, looking at how much neuroscience is taught in neuroscience degrees across the UK. Spoiler alert, very variable. So I started my lectureship in 19 and have kind of progressed through that. I'm now a reader and also an associate dean as a secondment at the moment. But looking at this timeline, there are lots of times where I have sought to accredit the teaching that I've been delivering. And I think it can be really difficult to get access to teaching opportunities. Certainly working with lots of students, they often find this is a bit of a struggle. So these are accreditations via the Higher Education Academy or Advanced HE that they're called now, along with things like National Teaching Fellowships. So I'm going to delve a little bit into that during this talk, focusing on my teaching experiences. And although I did have some before I began my lectureship in 2019, they were very kind of lucky. I was kind of in the right place at the right time. But this is what my lectureship looked like in 2019, quite quickly. I mean, I had about 6 months before everything went like this onto Zoom. And actually, being greeted with 500 black boxes when you're delivering a lecture, fairly soul-destroying, actually, but a great learning opportunity. It's not all bad. And I was working with colleagues who'd been teaching for a very, very long time, and I think everybody was, floored by this. But actually, I think it presented a real opportunity for us as educators to think about how we teach and why we teach. And I was always astounded as a learner, but now as an educator, that we don't really need or require formal qualifications of our lecturers in teaching. I do lots of public engagement work, and lots of people are like, what training did you get? A school teacher requires a lot of training in school teaching, but we don't necessarily require that in university and higher education as well. So I took this as a little bit of a challenge to come up with some innovations in teaching. Now, the word innovation is used generously, but I've used it here. And this gives you an example of what my standard teaching environment looks like. So this is our biggest lecture theatre in Cardiff. Very steep, these steps here. This seats about 600 people. And it's really tricky, isn't it? Because what can you do with that lecture theatre? You can't move the furniture around. The learner experience is very different if you are teaching a lecture of 500 versus a seminar of 20, for example. So this can be quite challenging. And looking at the sector, we are moving towards larger class sizes, and that's something we're not really getting away from. So I thought, how can I make this a bit more engaging for people, online or in person? So we've done things like polling, Mentimeter, even things like well-being polling, just asking people how they are at the beginning of a lecture. That goes down really well. Oh, I don't know if this is going to work. There is a little soundtrack. I'll just give a little intro if that's okay. I can hear clicks at the back. So, you know, online there's that dead space when everyone's joining a meeting and it's all a bit awkward, no one knows what to do. So I thought during the pandemic, I'm not going to have this because everyone's a bit awkward. I am going to play some music because there's sound pedagogical evidence that this actually enhances the learning experience, helps knowledge retention. And then it evolved into this thing where I was asking for requests from my students. But they had to be songs that in some way related to the content of the lecture. So I give lots of lectures on cardiac muscle. Does anybody want to guess what the song was? If we can't get it, it's okay. So it had to in some way relate. Yes, please. My Heart Will Go On, Celine Dion. It's a great guess, but it's not right. And I was a little bit worried about, you know, my love of kind of late 90s, early 2000s pop music coming through for my students. That's okay, authentic. Anyone else? It's a great first guess. Anything heart-related? Come on. Yeah. Staying Alive. It's a great guess. Yeah, I'll add these for next year. No, it's not right. Yes. No, it's a great guess, Tara. No. No! Yes. Sorry, break your heart. Unbreak my heart. No. No other guesses. Have we got it? We haven't got it? Is it going to work? I've built it up now. Does that help? It's Bonnie Tyler, Total Eclipse of the Heart. Thank you. A bit of a Welsh theme there too, which is nice. We can, yeah, thank you. So, yes. So this was just a way of making it a bit more engaging, goes down really well, and just kind of thinking outside the box. So things like sharing pronouns as well, I feel quite passionately about, and encouraging people who are teaching to be authentically themselves in the way that they teach as well. That's really important to me. Also, love a prop. I didn't bring any props today, but things like these bricks that are actually made of foam. I do a very good acting job in some of my lectures on motor control. When I was given a lecture on osmolarity and tonicity, I was worried. How was I going to make that engaging? You can put gummy bears in different isotonic solutions. This is an exploding bladder, practical, looking at smooth muscle control, some draw-alongs, things like this. But I am always really interested when we are conveying information, be that through teaching or science communication or outreach, how we work out who is in our audience, and therefore we can adapt and modify the information for them and just make it fun and interesting and engaging, because I'm sure we've all had that experience of maybe less than inspiring teaching experiences. So if I can help people learn in a way that they'll remember, then that's kind of my job, or I see it as my job. Some other props here, just to give you some examples, spent a lot of time painting shoeboxes for nucleotides. But over on the right here, some examples of storytelling. So the power of storytelling is really important to me. So this picture of my grandparents who were impacted by dementia, and I talk a lot about my experiences of that. Very mindful, as kind of Tara mentioned earlier, you know, dementia is such a common condition, but also when you have such large class sizes, you just have to be very aware. You don't know who's in that audience. People will be impacted by all sorts of conditions. And then, of course, my cats, Peter and Peggy, looking at things like neuronal control of stepping and motor control as well. So yes, my cats make regular appearances. They made even more regular appearances on Zoom. Yes. One thing I wanted to mention was the pastoral support. So I don't think we can talk about teaching and education without acknowledging the pastoral support that we as academics are required to give. I think it's such an important piece of the puzzle in terms of the student experience, but I also think people who are delivering teaching find this incredibly difficult. It's a major source of anxiety. So how we can support learners and students, particularly in these really challenging times. We know things like mental health are impacting studies. Lots of our students are balancing their studies with part-time jobs, for example. So how can we provide the pastoral support and ensure that people who are not, qualified therapists can provide that? So it is an important part of the puzzle. We also know that there are huge ED&I implications to this around who is doing this type of work as well. So there are unequal gender distributions there as well. But I wanted to mention it because I think it's really important, but it's also a real opportunity to potentially really change things for people by helping them and just taking the time to do that. But I do know that it is a source of lots of anxiety for staff. Oh, the tissues and the cheerleading. Yes, so although this work can be incredibly challenging, and I will be the first to admit that I've personally lost a lot of sleep over this, the cheerleading aspect and the mentorship and the guidance I see as an incredible privilege to be a very small part in that puzzle of helping people not only understand, the knowledge and the information and the complexities of neuroscience, but actually breaking down some barriers and discussing that wider pastoral piece too. Widening participation. So for a long time, I was on the admissions team, a job that many an academic will run away from. But I think that widening participation piece, who are our learners, understanding who they are, why they're coming to university is really important. You'll see some parallels here, because I think there are parallels between things like outreach, the pastoral support I mentioned earlier, but also employment opportunities. As academics, we're regularly asked for things like career advice. Many of my tutees go on to an incredibly awesome, amazing range of careers, whether that's in science or education or science communication, or banking seems to be quite common at the moment. I will happily write those references because I think the transferable skills are numerous. But it's thinking about and helping people to consider all of the options that they have available to them. I wanted to put a little slide on nerves, confidence and imposter syndrome because I remember giving my first undergraduate lecture, and it was in person, to probably about 100 students, thinking, oh my goodness, I'm going to be found out. Who am I? I don't know anything. This is terrifying. No one's told me how to give a lecture. I'd sat through hundreds of lectures as a student, but nobody had said, this is how you give a lecture. So I think that's where there's potential value here. And actually, for me, this comes back to authenticity, and there is sometimes a perception that we as lecturers know everything. It's not true. And I'm very happy to talk to students about the things I don't know about and come back with answers, etc. too. Because for me, that gets down to some of the kind of unhelpful power dynamics that we sometimes have in academia as well, which align with some of those pastoral concerns too. So that's my kind of whistle-stop tour. A few things in terms of where to start. If you haven't heard of the Advanced HE Accreditation Programme, that's already in existence. So this might vary if you are at an academic institution. Some people have like internal processes for this. Others don't. You can go direct to them. So they do Associate Fellow, Fellow, Senior Fellow and Principal Fellow. So this is a lovely way of accrediting that teaching, as I said earlier. So many of you will have great teaching experiences, which you can put on things like CVs and cover letters, etc. But to also say your teaching's been accredited is quite valuable as well. But I do think there's potentially a role for the BNA to play here, particularly for neuroscience teaching too. Oh, there they are, the different categories. And there is an online tool as well. You can go online and put your background in and it will highlight a particular category for you. So if you're unsure, go and check that out. Lots of people massively underestimate what they need as well. They think, oh, that wouldn't be for me, I haven't got enough, etc. Just have a look. Please don't underestimate yourselves and rule yourselves out before looking at that. So that's just a where to start slide. This is a lovely quote that I like to end most of my talks with. By being yourself, you put something wonderful into the world that wasn't there before. I think this is so important in teaching and education because we have such an opportunity to inspire the lives of literally thousands of young people. And I feel really, really grateful for that opportunity. So I will end there and say thank you. Very happy to take questions.
Thank you, Emma. Any questions? Yes, please.
I think it'd be really, oh, thanks. So teaching stuff that you are an expert in or that you really enjoy is great. It's really nice to get students excited. But often we have lack of resources and we end up teaching things we don't necessarily want to teach or we don't feel comfortable with. So how do you approach getting excited about and developing teaching on topics you're not really passionate about?
Yeah, it's a great question, isn't it? And I think particularly with the climate of lack of resource. So I do lots of team teaching. I think we have a tendency as academics who are trained in a niche area, understandably, to want to teach on that, but we can, extend that absolutely. So I think number one is having the time to be able to develop that confidence, reading around the subject, etc. But then I think about, what learning outcomes do I want my learners to go away having understood, and how can I convey those in a way that isn't just me talking at a screen? So is that a prop? Is that a story? Is that an analogy? And actually, it depends on the level, absolutely. But at Cardiff, we have a core first year. So we have students who are studying lots of different programmes. And actually, developing a lecture for first years in particular, many of whom are just out of school. So we all have such a great background and training that you can absolutely do that. So it's something that I do completely acknowledge. I work with lots of colleagues who are scared, worried about that specialism and extending that too far, perhaps. But I would just, challenge people to say, let's just broaden that out a little bit. Yeah, because it's a question of why, isn't it? Why people don't want to push that out. And I think you can absolutely push that too far. But the question is, how far do you push it, isn't it?
Hello, that was very interesting. Yeah, you spoke about the sort of diverse amount of training that people have in order to start doing their neuroscience teaching. But there's also quite a lot of diversity in the type of teachers that we have, and that people who find themselves doing neuroscience teaching have. So people have lots of different priorities. Some people have jobs where they do teaching full time. Some people would rather get back to their other things in the lab. How do you get the best out of those people, and how do you get the best out of the people who maybe don't want to teach at all and maybe have to? And how do you get the best out of people who would love to, but maybe that isn't their sort of major priority?
Yes. Again, a contentious issue, isn't it? I think that across the UK we have a real problem with finances at the moment. And when we look at what pays the bills, it's teaching. And I do acknowledge that there are people that don't want to do that. And there are people that are kind of bought out of teaching sometimes. Interestingly, we don't get that research buyout. So, sometimes I've been offered to be bought out from teaching to do admin roles. But for me, and this, I completely acknowledge this isn't the same for everybody, it's what gets me out of bed in the morning, the fact that I can help people understand a topic. So I think it is about understanding your team and the skills that they have. But I also think this needs to come from senior leadership around, actually, if we are an organisation that teach, we have to not only teach, but do a quality and good job of it. So, I'd like to think we're moving away from a time where we could get away with somebody stood at the front and just talking off the screen for 50, 60 minutes, because that's not actually helping anyone. And then I would be keen to explore why people don't like teaching, because I acknowledge there are different priorities. For me, we've done a lot of work around the reward and recognition piece as well. So in terms of reward and recognition of teaching a scholarship, it's a much greyer area than the reward and recognition of research. So researchers, publications and grants, tick, tick. Scholarship is a much more diverse area of specialty, but I think nonetheless an important one. So yeah, I would challenge that perception of, you know, do you just not want to teach or can't you teach? Because I think everybody can and should be doing a good job of it too.
Any other question? Yes, please.
Thank you. I don't know if this is the case everywhere, but I think it's very common that PhD students don't really have many opportunities other than demonstrating, which is not really teaching, really. So do you have maybe any advice on how we could get more experience in the area?
Yes, so I think demonstrating is a lovely first step. So I think within demonstrating, I'd break that down into different opportunities. So certainly the demonstrating I did, there were opportunities for kind of one-to-one interaction with students, so which is almost a bit tutorial-ish, but then much bigger opportunities with very, very large practical classes, I can remember too. So I think it is about talking to academics. So it's really interesting, isn't it? Because I think the academics, and particularly those who have very heavy teaching loads, are absolutely overwhelmed. We've got early career researchers and PhD students who are saying, I'd love some more opportunities, but that isn't quite linking. And I think a lot of that is absolutely about quality. We have to ensure that the teaching is of good quality, but I absolutely have confidence that those PhD students can deliver quality teaching too. So I think a nice middle gap there is things like tutorials, workshops. So I think it's about talking to academic staff as well. So my first lectures that I gave were when somebody was ill. So at quite short notice, there were 3 lectures to give. They allowed me to do it on my research area at the time was Huntington's disease. So I was like, great, 3 hours to talk about my PhD, amazing. So yeah, I would talk to academic staff about that. And then I would also talk to if there's like a postgraduate department as well, because lots of universities are being much more strategic now because we do have those teaching gaps about how we can utilise those.
If I can add to that, I think supervision of undergrad students is also teaching, and that needs to be recognized as well and can be put on a CV as teaching experience. So that's another way to get into teaching.
Yeah, absolutely.
Yes, another question there.
Thank you. That was so interesting and inspiring, even though I'm not an academic and don't teach at all myself. So this may be a really ignorant question, but I'm wondering about the less fun aspect of teaching, which is marking. And to what extent is that something that puts people off in terms of the workload? And can there be training in that? My sister-in-law is a PhD student and does a lot of marking and has had 0 training, which can also put people off.
Yeah. Absolutely. So there are no ignorant questions. Thank you. I think it's a great question. I was almost a resounding when you mentioned marking, wasn't there? So yes, at Cardiff we actually approach our PhD students to help with the workload of marking. So that might tie into your question earlier. Training for marking, absolutely, but I also think we need to be a bit more clever about how we design our assessments, so that we don't set ourselves up to be marking 200 essays in a dark room. You don't have to do it in a dark room, obviously, but yeah, so I think marking is important, but I also think this is a personal view, probably over-assess. So training in marking, model answers, etc. There's some really interesting pedagogic research on variability of markers, particularly if you have a large student cohort and you might have a marking team within that. But I don't think we shouldn't be doing teaching because the marking's a bit less exciting. Can we make marking exciting? So things like group marking, round table marking, it's not quite as exciting, but it just prevents it's not quite as awful. If you do it in a team, marking circles can be quite useful because it's not quite so isolating. But I would also add, I think the design of the assessments is really important because we see massively unequal marking distribution between teams. So we know that women, for example, often have a significantly higher marking workload if you're giving students optionality within the questions. And I'll leave it.
Any other questions? Okay, so thank you, Emma.
You're welcome.
So just a short conclusion to this session. So as you've seen today with 3 different taster sessions, this Brain Training Academy is offering a range of courses designed to support neuroscientists at all stages of their careers, whether from undergrads, postgrads, master students, PhD students, postdocs, junior PIs, senior PI entering a new area of research. This is designed to cover all the career stages. Our training sessions do provide CPDs. If that's something of interest to you, do talk to us about it. I think at the moment not all of them provide CPDs, but if you're interested, we can develop that further. And BNA members do receive discounts on the course fees, and some of them are free when it's an hour online. So do join different future sessions to try this. So just to remind you, the Dementia Masterclass registration is open at the moment, and there are spaces available, so on the 4th of June in London, and we've seen how passionate Tara is, and so the day is promising to be really, really interesting. Thank you, Tara. And last thing is the QR code to provide us some feedback. So do tell us where the training gaps are for you, for your students, for the PhD students, postdocs in your labs, for the technicians, junior PIs, or even senior PIs, as I said. Do suggest ideas of trainings, but also help us deliver some of these training. If you have an area of expertise that you think would be helpful to some of the students, postdocs, PIs, do come and tell us or fill out that form so we can plan a session on some of these topics that you're expert in and that you can provide training in. So if there are other questions, we'll all be at the front here to speak to you. Otherwise, we'll just finish a little bit early this session. And thank you very much to all attending this session.